Hello students, I am teacher Svetlana, and in our today's lecture we will learn about gametogenesis, which is covered in the chapter Reproduction in Lower and Higher Animals. So let's begin. Gametogenesis. The process of formation of gametes in primary sex organs is called as gametogenesis. The conversion of spermatids into sperm is called as spermatogenesis, and the process of formation of mature female gamete is called as oogenesis. So gametogenesis includes spermatogenesis, that is spermiogenesis in male, and oogenesis in female. Have a look at this image here. It describes to you what is the process of gametogenesis, where in this process there is conversion of immature germ cells, that is spermatogonium or oogonium, into mature gametes, that is sperm or oocytes. This maturation is called as spermatogenesis in male and oogenesis in female. During gametogenesis, a special type of cell division, that is meiosis, occurs, in which the number of chromosome is reduced from diploid to haploid number. This prepares the gametes for fertilization, that is the formation of sperm, and the formation of the ovum takes place. So we will see this processes in detail in this lecture. Students, let us first have a look at spermatogenesis. The process of formation of the male gamete or the sperms or the spermatozoa from the germinal epithelium of testis is called as spermatogenesis. At the onset of puberty, the hypothalamus begins secretion of gonadotropin releasing hormone. It initiates the significant increase in the secretion of follicular stimulating hormone, which induces spermatogenesis. Each semiferous tubules is lined by a single layer of cuboidal epithelial cells called as germinal epithelium. Now students, these cells of the germinal epithelium undergo spermatogenesis to produce sperm. The process of spermatogenesis involves three phases, namely multiplication phase, growth phase, and maturation phase. Let us have a look at each of these phases. The first phase is the multiplication phase, where the male germ cells, also called as the spermatogonia, undergoes mitotic divisions to form large number of spermatogonia. So what happens in the multiplication phase? Have a look at this image here. You can see the initial primordial cells. So these primordial germ cells, which are 2n of the semiferous tubules, they undergo repeated mitotic divisions and produce large numbers of these spermatogonia. So this is due to mitosis, you have these spermatogonias that are produced. This is the first multiplication phase. Each spermatogonia is diploid and with 46 chromosomes. Then the second phase is the growth phase. The spermatogonia increases their size by accumulation of nutrients in the cytoplasm and are ready for meiotic division. And the spermatocytes are called as the primary spermatocyte, which have 46 chromosomes. So here, have a look at this image here. This is your second phase, that is the growth phase. Some of these spermatogonias, they stop dividing and they grow in size. So they grow in size and they develop into a primary spermatocyte, which is diploid to N due to accumulation of food. Then students, the next phase is the maturation phase. The primary spermatocyte completes the first mitotic division, leading to the formation of two equal haploid cells called as secondary spermatocytes, which have only 23 chromosomes each. So if you have a look at this image here, this is your third phase, that is the maturation phase. The primary oocyte undergoes the first mitotic division and it produces the secondary spermatocytes, which is diploid in nature. So it has only 23 chromosomes. Each of this secondary spermatocyte undergoes the second meiotic division to produce 4 equal haploid spermatids. So then they further undergo the next step of meiosis 2, and they are now developing into 4 haploid spermatocytes, which are your spermatids, and they have equal amount of chromosomes which is in the haploid state. Then these spermatids that are formed, they are non-motile and they are non-functional. They get transformed into functional spermatozoas by a process called as spermiogenesis. So what is spermiogenesis, students? The spermatids are transformed into sperms, which is also called as the spermatozoa by the process of spermatogenesis. After spermatogenesis, the sperm heads become embedded in the serotoli cells and are released from the seminiferous tubules by a process called as spermination. Now, during spermatogenesis, when your sperms are getting formed, there are certain changes that happen. That is, the length of the spermatids increase, the centrioles are rearranged as primary and distal centrioles, mitochondria become spirally coiled, and acrosome is formed from the Golgi complex. So these are the changes that happen during the spermatogenesis, and after the sperms are formed, then they are released from the seminiferous tubules. Now students, let us have a look at the structure of the sperm. It is microscopic motile structure composed of head, neck, a middle piece and a tail. Have a look at this image here. This is the head region, then you have the neck region, then you have the middle piece, and this entire is the tail region. The whole body is covered by plasma membrane. Now, the head of the sperm is oval in shape and it contains haploid nucleus. Above the nucleus, there is a cap-like structure which is called as the acrosome. So, the sperm head contains an elongated haploid nucleus and the anterior portion is covered with a cap-like structure called as acrosome. Here, this is your acrosome. Now, this acrosome contains hydrolytic enzymes like hydrolyase and proteolytic enzyme like zona lysine and corona penetrating enzymes. Now, so these enzymes are present which helps the sperm to penetrate into the ovum. Then we have the neck region. This is a very short region having two centrioles, that is a proximal centriole and a distal centriole. The middle piece contains numerous mitochondria which produce energy for the sperm motility needed for the fertilization. So the middle piece has an axial filament surrounded by around 10 to 14 spiral turns of mitochondria. Here, this is your middle piece. This produces the much required energy which is necessary for the movement of the sperm. The tail helps the sperm to swim to reach the egg cell. So this tail is helping it to swim and reach the egg cell. Now, the tail, it is a long, slender and a tapering part containing cytoplasm and fine thread-like axial filament. The axial filament arises from the distal centriole and travels throughout the length of the tail. It is partly surrounded by plasma membrane. The part without the plasma membrane is called as the end piece. This is located here. This is the end piece. Now, the seminal plasma along with sperm constitutes the semen. Now, students, let us have a look at the process of oogenesis. Students, the effect of gametogenesis in females is associated with the mature female gamete. This is created through a process called as oogenesis. This happens in the ovaries or the female gonads. There are three phases to oogenesis. Just as we have in the spermatogenesis, this process is divided into three phases, that is the multiplication phase, growth phase and maturation phase. Let us try to understand these phases in detail. The first phase is the multiplication phase. Have a look at this image here simultaneously. This shows you the first phase, that is the multiplication phase. In this stage, the primary germinal cells, PGC, which is diploid 2N of the ovary, undergoes repeated mitotic division to form millions of gamete mother cells or oogonial cells. So it forms the oogonial cells which are again diploid. This process is completed in the embryonic stage of human females. That is, during fetal development, it is noticed that certain cells present in the germinal epithelium of the female ovary are bigger than the others. Hence, these cells split by mitosis, creating a couple of million oogonia, or the mother egg cells, in each ovary present in the fetus. There are no more oogonia which are formed or augmented after birth. The second phase in oogenesis is the growth phase. Now, some of the oogonia stops division and begins to increase in size. Have a look at this image here. Some of them stop the division and they start becoming larger in size, and they form the primary oocyte, which is diploid to end. Cellular organelles like endoplasmic reticulum, Golgi apparatus, and mitochondria also increase in number. So in this phase, this particular process or procedure of primary oocyte tends to be very long. In this, the oogonium nourishes into or nurtures into a bigger primary oocyte. After this, each primary oocyte gets surrounded by a granulosa cell layer to create the primary folliculi. A large number of folliculi get debased during the duration from birth to puberty. Therefore, at puberty, around 60,000 to 80,000 primary folliculi can be found in each ovary. So here, this is your second growth phase, where you have some amount of oogonia developing into the primary oocyte. Then the third stage is the maturation stage. Similar to the primary spermatocyte, every primary oocyte experiences two maturation divisions. However, the outcomes of the maturation division under oogenesis are quite different to those which occur in spermatogenesis. Considering the first meiotic division, the primary oocyte segregates into two uneven haploid daughter cells. These are known as the large secondary oocyte or the ovum and a small polar bodies. So this is your first meiosis. And then this is your second meiosis, where you have a large ovum and two polar bodies that are formed. The ovum so formed functions as the female gamete and it is ready for fertilization. Completion of meiosis 2 and completion of fertilization goes hand in hand. If the secondary oocyte does not receive the sperm or the spermatozoa, it is shed off along with the menstruum. Now students, let us have a look at the structure of the secondary oocyte. Ovum refers to secondary oocyte stage of oogenesis, where the second maturation division is yet to occur. In this condition, the matured ovum is released from the ovary and it enters into the uterus in mammals for fertilization. This here is the image of the unfertilized egg or the ovum, which is also called as our secondary oocyte. Each ovum is rounded and is non-motile structures. The cytoplasm of the egg is called as ooplasm. The peripheral layer of ooplasm is known as cortex and it contains many microvilli and cortical granules. The nucleus is large and is centrally placed in mammals, but towards the animal pole is the yolky egg. So here you have, this is the egg region, and here this is called as the animal pole, where you have the yolky egg. Ovum is covered by thin vitelline membrane, which further covered by another primary membrane known as zona pellucida. So here you have, this is your vitelline membrane. There is a narrow space between the two membranes called as previtelline space. This space is called as the previtelline space. During discharge of the ovum from the Graafian follicle, several layers of epithelial cells adhere to the outer surface of zona pellucida and are arranged radially, forming corona radiata. So this is your corona radiata here. So this is how the unfertilized egg or the ovum looks. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.